Thanks for coming by. And what I want to do for just a few minutes is explain how to use a Punnett square and how to interpret what is in a Punnett square once you've used it. And to do that, we also need to throw around some words that you may not be familiar with, but that I use them a lot when asking questions about Punnett squares and analyzing and interpreting Punnett squares. One of those words is genotype, and the definition we used in class is the genetic makeup shown by the letters. Some of the genes that you have, for some of the traits that you have, some of the characteristics that you have, we can represent those with letters. And it doesn't really matter what letters we choose to represent those traits. You know, maybe it's the letter T or the letter D or something like that. You know, the genotype is simply the letters. The phenotype is the physical trait shown by the genotype. That means it's what the letters represent. You know, what does big T represent? You know, what trait does that represent? What does that look like if we could see that particular characteristic of that person or thing? Big D, little d. What characteristic does that represent if we could see that in somebody? Which means we would need to know what the big D stands for and what the little d stands for and stuff like that. And we'll get into all that stuff. Heterozygous is an organism with two different alleles for a trait, which means like one, you know, in just simple layman's terms, that means one letter is capital, one letter is lowercase. You could also write it like this, but that's the same thing. And it's just kind of standard convention, you know, standard practice to write the capital letter first. But heterozygous means one's capital, one's lowercase, and these represent two different forms of a particular gene. And then that leaves us with homozygous. Homozygous is an organism with two alleles for a trait that are exactly the same, like two capitals or two lowercase. And again, the letters represent forms of a particular trait, like whether you can roll your tongue or not, whether you have free earlobes or attached earlobes, which way your hair spins around the little spot on the back of your head, if it goes clockwise or counterclockwise. Maybe clockwise is capital, counterclockwise is lowercase. So this would be two clockwise, this would be two counterclockwise. This one has one of each. It's heterozygous. Homozygous, they're the same. They're either both the recessive form or both of the dominant form, which is something else that I don't even have on here that needs to be mentioned, is dominant recessive. Dominant recessive means that if a trait is dominant, that trait hides the recessive form of that gene. That means if you inherit both, like this person, the dominant, which we represent with a capital letter, that this is going to hide whatever that is. And so this person is going to show whatever this trait represents. Maybe this means they can roll their tongue. And so maybe they inherited both forms, you know, can roll your tongue and cannot roll your tongue from their parents. And so the big T means, well, this gets hidden and they can roll their tongue. And we'll talk a whole lot more about that in a minute. So if you're still kind of confused by all this, that's okay. When we get into it, I'll explain it some more. And so when working with the Punnett square, you need to know what the parents are. And sometimes half the trick in working a Punnett square is deciphering the problem to figure out what the parents are. Because it may use words like homozygous, heterozygous, genotype, phenotype in the description of the parents. And so you need to know what those mean and how to figure out what the parents are. In this case, I just flat out told you they are big T, little t. And what we are saying that the T stands for is we're just going to go something really ridiculous and say that it stands for tails. And so a capital T means no tails, which that makes sense since most people don't have a tail. We're going to say that the capital letter, which will be the dominant form, is no tails, and that a lowercase t means that you have a tail. And so both parents have both forms of this gene. Now, the big T is dominant, So it's going to hide the little t, and that's going to mean that this parent does not have a tail. The big T is dominant. Capital letters always dominant in these examples, which by the way are called simple dominants. In these examples, the capital T is dominant, and so this parent also has no tails. So both parents with no tails. Now, what we can do, since we know the letters or the genotype of each parent, that's their genotype. Their phenotype is no tails, and the phenotype of that parent is no tails. So genotype is the actual combination of the letters. The phenotype is what that would look like. Their genotype is big T, little t. The phenotype is no tails. All right, since we already know this about the parents, then what we can do is use a Punnett square and make some predictions about the offspring. And so what you're going to do is take one parent, and it doesn't really matter which one, I just picked the first one, and you put one letter of that parent above each column, just like that. So big T, little t is that parent right there. This parent goes on the side, and it's one letter per each row. So big T, little t. So here we have big T, little t, that's one parent, and then our other parent is over here, big T, little t. Now here's how to use the Punnett square, and this gets kind of messy, in my opinion. In fact, let's turn this off from full screen for just a little bit. What you're going to do is take the letter that's on top, and it's going to go in every box down below it. So this big T is going to go here, and it's going to go here. All right. This letter also goes in every box below it. Little t, little t. Now you might remember that I said that I prefer to put the capital letters first. That's just kind of the standard way that it's written. And because of that, I kind of offset my little t's over to the right a little bit for that reason. All right, well, let's use that giant eraser, make it not so giant. And let's erase these lines. We don't need the lines. And once you're comfortable using Punnett squares, I wouldn't use the lines because it just clutters things up, makes things harder to read and interpret. But for now, I want you to see where these letters in these boxes come from. So over here, we've taken care of the top parent, the parent on the side. This capital letter or whatever letter is out here, doesn't have to be capital, goes in every box out beside it. So it goes in this box and it goes in that box. And so that's going to be that big T is going to go there, and this big T is also going to go here. We're going to do the same thing down here. This letter that's out to the side goes in every box out beside it. It's fairly simple, right? So that's a little t, so a little t, and a little t is going to go here too. And let's erase all these crazy lines.
During the course of the video, you may notice that the video speeds up, and that's so we're not wasting so much time, watching me draw and erase.
So again, very simply, it's you take the letter that's on top of the column, it goes in every box below it. The letter that's on top of the column goes in every box below it. The letter that's to the left of the row goes in every box in that row. And the letter that's to the left of the row goes in every box to the left of the row. That's where these letters come from. All right, now I'm going to attempt to select all this stuff. So we have the Punnett square, but so what? So let's try to figure out what is going on here. And I don't know if this is going to work or not, but I'm going to try it. All right, so here's our Punnett square, and these are questions that I would typically ask with a Punnett square or when solving a Punnett square. And in our Punnett square, because I didn't mention this earlier, these represent the possible offspring of these two parents. Doesn't mean they're going to have 4 kids because there's 4 boxes. That's not what that means. This means that every time they have a kid, it can be this one, or it could be that one, or it could be that one, or there could be that one. If they have 4 more kids, or 4 more kids, well, okay, they could have 4 kids. If they have 4 more kids, every one of them could be that one right there. It just means that every time they have a kid, these are the 4 possibilities. So the question is, how many genotypes? Well, we have 1. Genotype means the combination of letters. How many different combinations of letters are there in the possible offspring? We have 1 that is 2 capital T's. All right, so there's 1. We have a capital T, lowercase t. We have capital T and lowercase t. Well, these 2 are the same. All right, so that's the same genotype. And then we have 2 little t's. That's a different genotype. So we have 1, 2, 3 different genotypes. 2 capitals, capital lowercase, 2 lowercase. Okay, the next question, what are the phenotypes? Well, big T, big T. Big T stands for no tails. They got a big T from each parent. So the phenotype is no tails. Big T is a genotype. No tails is what they would look like. The phenotype for this one, big T and a little t. Remember, the big T is dominant. It hides the lowercase letter. Big T stands for no tails. So this person is also going to be no tails. This one, like we said a second ago, is the same as this. And so this one is also no tails. We'll go ahead and write it in there, although it's not really necessary. And then the phenotype of this one doesn't have a capital T in it. It's 2 lowercase t's. The phenotype for this one is tails. The genotype is little t. Phenotype, they have a tail. So those are our phenotypes. And I don't really have room to write it up here. So that's kind of why I wrote it in the box. What percent would not have tails? Okay, well, there's 4 boxes. And 1, 2, 3 out of the 4 would not have. 3 out of the 4 would not have tails. So that is 75%. That is the worst looking 75% that I've written in the last 5 minutes. Now the next question, what percent would have tails? Well, we have this one, and that's 1 out of 4. 1 out of 4 is 25%. Now we have 2 more words that we really haven't said a whole lot about. What percent are heterozygous? Heterozygous means that you have a different, 2 different forms of the same gene. In this case, we're pretending there's a gene that makes tails. And so what percent have both forms? The simplest way to think about this is which ones have a capital and a lowercase. That's what heterozygous means. And so this one has a capital lowercase, check mark. This one has a capital lowercase. These 2 are heterozygous. So that's 2 out of the 4. 2 out of 4. We also like to call that 50%. And then the last one, homozygous. Homozygous means the 2 letters are the same, or it means they have the same form of the gene. Got it from both parents. But like I said, the simplest way to think about it is they're either both capital or both lowercase. That would be this one, because it has 2 capital letters, and this one, 2 lowercase. Again, that's 2 out of the 4, which we like to call 50%. All right, so we have genotypes, phenotypes, heterozygous, homozygous, dominant, recessive. We have all these vocabulary words that go along with the Punnett square, but this lets us interpret possibilities for the offspring. If you have questions, of course, you can always rewind the video to different parts or watch the whole video again if you need to. But there you go.